everyone, my name is Jiumei, who I am a PhD student from the Johns Hopkins University. Today I'll present my research on developing a new immunoassay called MagDroplex for highly sensitive, multiplex, and accessible digital protein measurement. So a lot of protein biomarkers in the body fluids, such as the peripheral blood, are presenting at low levels under the pathological or physiological conditions, therefore requiring highly sensitive methods for detection. Meanwhile, the multiplexed protein detection, which allows the detection of multiple protein targets in a single reaction, has gained much attention because it can speed up diagnosis, reduce assay cost, and also provide a comprehensive view of the biological conditions that involve multiple biomarkers. So here I want to give some examples. First, in the field of cancer diagnostics, the well-known liquid biopsy test, which is the cancer-seq, requires the sensitive detection of multiple protein biomarkers in the blood, facilitating the early identification of multiple cancer types. And in the diagnosis of neurodegenerative biomarkers that typically involve multiple biomarkers, there's a growing interest in detecting these biomarkers in the peripheral blood, which is less invasive than sampling the cerebral spine fluid. However, when these biomarkers pass the blood-brain barrier, the concentration can decrease sharply, therefore requiring highly sensitive methods for detection. So the enzyme-linked immunosorbent assay, or ELISA, remains to be the current gold standard for quantifying the proteins. The ELISA is typically done on a microwell plate. So the diagram here shows the working principle of one classic ELISA format known as sandwich ELISA, which converts the detection of protein targets into enzyme-catalyzed signals that indicate the abundance of protein target in the sample. So the ELISA is widely used in both research and clinical settings. However, the sensitivity is only limited to picomolar level, and multiplexing in individual well can be very challenging. Sorry. So to overcome the limitation of ELISA, researchers have developed a new method called digital ELISA. So instead of running the assay in a microwell plate, digital ELISA uses magnetic beads to capture the protein target in the sample and then form an immunocomplex on the bead surface with the enzyme labels. Next, the beads are digitized into femtoliter-sized microwells, where the enzyme label can catalyze the substrate to generate highly concentrated fluorescent signals in the well. So because digital ELISA is measuring protein target at the single molecule level, it can achieve exceptional sensitivity down to the femtomolar level. And the multiplexing can be achieved by using color-coded beads. But right now, the most common multiplexed digital ELISA format is only 4-plex. Further increasing the multiplexing level can be very tricky, which is constrained by the capacity of the microwell array to accommodate more beads. And moreover, digital ELISA lacks broad accessibility due to its reliance on complicated instrumentations. So our aim is to develop an immunoassay that can achieve combined advantages of high sensitivity, scalable multiplexing capability, and broad accessibility. So our assay is called MagDroplex. So it has a workflow with 2 main stages, including the magnetic bead-based proximity extension assay, or MagPEA, that converts the detection of protein target into the DNA templates. And the downstream fluorescence coding droplet digital PCR that can identify and quantify these DNA templates. So among the 2 stages, the DNA are released from the beads by heating, facilitating a bead-free droplet digital PCR reaction that can accurately measure every single DNA. So in the following slides, I will explain how our method can achieve these combined advantages. So first, our assay achieves high sensitivity because of 2 factors. First, it generates very low background signals in the MagPEA workflow. So here, in the MagPEA, we first use magnetic beads to capture the protein target in the sample, and then we add multiple pairs of PEA probes to further recognize this protein target. So the PEA probe here refers to the antibody oligo conjugate. So when two PA probes bind onto the same antigen, they are brought into close proximity, so the oligos can hybridize to each other and then being extended to generate the double-stranded DNA template. So here we can see a true signal requires 3 antibody bind onto the same antigen, and the beads can also enable extensive washings, so substantially reducing the background signal generation from any non-specific bindings. And the second, because our assay is converting the detection of single protein into single target specific DNA template, which is digitally measured in the downstream digital PCR. So it has single molecule sensitivity. And the multiplexing of our assay is achieved by multiplex PCR amplification. Specifically, we use a fluorescence coding PCR that we developed before on these DNA templates. So as I mentioned earlier, the detection of protein targets is converted into the detection of multiple DNA templates. So if we take a closer look at these DNA templates, each of them has a poly-T handle, have a universal primer binding sites, and a varying number of green or red Tech-Man probe binding site, as well as a unique hybridization site. So if we digitize these DNA templates into individual microreactors, so after the PCR, each of them can generate unique fluorescence signatures. So taking the third one as an example, so during the PCR, the Tech-Man probes can first bind onto the DNA template, followed by the binding of PCR primers. So when the PCR primer extends along the DNA template, the Tech-Man probe can be cleaved to generate fluorescence intensity at the 2 channels. So for a different target that has different number of green or red Tech-Man probe binding site, they will generate different endpoint fluorescence signatures, forming different color code combinations that can facilitate the identification of different targets. So right now, our assay uses the Bell-Red QX200 droplet digital PCR system to perform the digital PCR. So where the DNA target are digitized into around 20,000 droplets. So after the PCR reaction, the fluorescence intensity in each droplet are detected on the droplet reader, and this droplet can be clustered into different groups to facilitate the target identification and the quantification. However, our method is compatible with any commercial or custom developed digital PCR devices, therefore providing higher accessibility to the researchers that are interested in researches that require highly sensitive and multiplexed protein detection. So as a proof concept, we have designed the PA probes targeting 8 human cytokines. Specifically, the oligos on these PA probes have a unique number of FAM or hex probe binding site, which are labeled in green or red, respectively here. And for the single-plex assay of each target, as what we expect, we can see distinct endpoint fluorescence intensities for each target at the FAM and hex channels. These droplets can form tight droplet clusters in the different locations in these 2-dimensional amplitude plots. So we have also tested serum-diluted protein targets spiked in the phytobovine serum. So here, as we decrease the concentration of a target, we can see a decrease in the droplet digital PCR signal output, suggesting the effectiveness of our assay to quantify the protein targets in a sample. Moreover, our assay has achieved sub-femtomolar to attomolar detection sensitivities for all 8 targets that we tested. So next, to validate the capacity of our approach to perform multiplexed protein detection in a single reaction, so we have mixed everything together and then run the assay in just one well. So here, our results show distinct droplet populations in different locations with different fluorescence intensity at the FAM and hex channels. We then developed a data data analysis pipeline using the Gaussian mixture model. And here we can see 8 distinct droplet clusters that can represent each protein target. So our assay also shows pretty good specificity. And for each target, with the presence of all the bead and all the peer probes, the droplet digital PCR output is comparable to what we have seen in the singleplex assay, suggesting the minimum cross interference on the signal generation among all the tested targets. So we have also tested serially diluted protein mixture spiked in the phytobovine serum. So here, for each target, we can see pretty good linear fitting for all of them. And the limit of detection in the multiplexed assay were found to be comparable to what we achieved in the singleplex assay. So here comes to the conclusion. Here we have developed an innovative protein detection assay with combined advantages of high sensitivity, scalable multiplexing capability, and the broad accessibility. So we have successfully demonstrated an 8-plex assay with that can achieve sub-femtomolar to attomolar detection sensitivities with pretty good performance. So in the future, we will first explore higher level of multiplexing, first by designing fluorescence codes across additional fluorescence channels. So here, so right now we have already achieved 8-plex detection across only 2 fluorescence channels. So if we incorporate higher number of fluorescence channels, like 3, 4, or even higher, then the multiplexing level can be exponentially scaled up. And in the multiplexed assay, we also need to increase the number of microreactors. And since we are doing the droplet digital PCR, so I think this can be readily achievable. So I don't know what happened. We will also explore the application of our approach in various biological contexts, such as the neurodegenerative biomarker detection and in some diseases like cancer and autoimmune diseases. So with this, I want to thank to my PI, Professor Jeff Wang, for the guidance. Thanks to Dr. Zhang and Dr. Park on the pioneering work on the MagPA and the fluorescence coding PCR. I would also like to thank my lab mates that involved in this project, as well as the collaborators. So thanks for listening. I'm happy to take any questions.